Right. Thank you, everyone, again for joining this session of the Bioconductor Africa Seminar Series. We are very glad to have you on this call and have you joining to learn something about the GWAS data analysis using the Genesis package. This is part two of this seminar series, particularly the GWAS data analysis. We began last week on Wednesday, and we are continuing that today. So before I hand it over to Stephanie again to lead the session, I would just like to point your attention to the chat. I have posted the link to the Framer pad. Please take part on our roll call and icebreaker, which should now be on line 37. You can tell us your name, your organization, and what you hope to learn from this session. And before we also start, I would like to read a few housekeeping rules. It's important for us to know how to interact with one another during this call so that we can have a smooth and seamless session. So we do have a code of conduct, and if you'd like to read it in full, it's on line 15 of the Framer path that's been posted on the chat. But the TLDR of this is that we want you to treat people with the same respect that you'd like to be accorded to yourself. So please do not tolerate any intimidation, bullying, or harassment. And if that happens to you, please message me during this call and we will take the necessary measures. This call is being recorded as you have seen. So if you would like to anonymize yourself, please change the name that you've used as your username. You can also use an emoji or anything like that to anonymize yourself. And the notes will be taken on the Framer pad. Also, again, if you do not want to be recognized, feel free to anonymize yourself accordingly. We will remove AI note takers from this call, unfortunately, because for privacy and safety reasons. Also, we will be uploading this call on YouTube, so you can always get it there, and the notes will be archived on our Bioconductor Africa Seminar Series webpage, which you can always navigate and find them there. Again, if you've not joined our Zulip channel, please scan the QR code on your screen right now. And if you've not joined our mailing list, please do that as well so that you can always be able to get updates from our community and also learn of any new training opportunities that may be available. So I will stop sharing now and then introduce our speaker, Stephanie, who's going to continue with GWAS data analysis using the Genesis package. If you'd like to read her full bio, it's on line 42 of the Framer pad, but I will just read the first paragraph so that anyone who's new to this and has not come across her work may know what she works on. So Stephanie has a background in astrophysics and over 15 years of experience in statistical genomics and genetics. She has extensive expertise in analyzing large-scale genomic data sets and developing scientific software, and is the author of several R and bioconductor packages and Python-based pipelines for phenotype-genotype association testing. Again, thank you everyone for joining. And now I will hand it over to Stephanie.
Hello, everyone. I'm going to share my screen. So last time we talked about association testing with Genesis, and we had not yet introduced including relatedness and population structure into our models. So that's where we're going to start out with today. I'm just going to briefly introduce the R packages that we'll use for kinship and population structure. The first one of those is, unsurprisingly, Genesis, which was written by Matthew Konemos. And it has an iterative process to separate recent familial relatedness from distant genetic ancestry. And so the process that it goes through is it first uses an algorithm called King Robust to get a set of initial kinship estimates for everyone in a sample. It then uses a process called PC-AIR to select an informative set of unrelated samples. And by informative, we mean people who are most representing the ancestral diversity of the sample. Um, so if you have a pair of individuals in your sample and one of them has a particular ancestry that's the most different from everyone else in the sample set, then that person would be selected in the unrelated set. And then PCR will do principal component analysis only on the unrelated samples, but then project the relatives to get PC values for them as well. And then the PC relate algorithm estimates kinship coefficients, but adjusting for population structure and admixture using the principal components. So this is a flow chart of what that process looks like. So we start out with the king kinship estimates. We get an unrelated set. We use PCA. We use the unrelated set for PCA. We get kinship estimates using those PCs, and then you can go around this circle again, taking those new kinship estimates you got, using those to get a new unrelated set, running PCA again, and then running PC relate again. Um, it's usually sufficient to just go around this circle twice. We don't generally go around more than twice. And if you have a very large sample set where it might be computationally intensive to do this, you can just do this process once and not iterate multiple times. These are the key functions in Genesis that we're going to see in the R code that we're going to look at today. Uh, The next package for population structure that we'll use is SNP-relate, and this package was written by Zhuwen Zheng, who's also the author of the GDS format and seq array packages. And this has functions for doing linkage disequilibrium pruning, PCA, identity by descent analysis, as well as computing a genetic relationship matrix. And so a genetic relationship matrix is a single matrix that is representing both the kinship and the population structure in a set of samples. So rather than Genesis, which splits out those two things into separate components, a GRM will attempt to represent the relationship between the samples in a single matrix. These are the key functions in SNP-relate that we're going to use in this session. And we'll talk more about these when we get to them in the R code. And then I just wanted to remind everyone of the essential R objects that we're going to use in this session. These are the same as the ones as what we saw last week, where we're going to construct seq_var iterator objects in order to iterate through blocks of variance in our GDS files. So now let's go over to the R code. So the first example that we're going to go through is just to do the simplest way to get kinship, which is just to compute a genetic relationship matrix using the SNP-relate package. There's multiple different algorithms that you can use to do this, and if you're interested, you can look at the papers that are linked here to learn more about those different algorithms and what the pros and cons are for those algorithms. A pretty common one is to use the GCTA method, which is what we're going to show here. So we load the seq array package, we open our GDS file, we load the SNP-relate package, and then we just call this function snpgds grm, which computes a GRM on all of the data in our GDS file. And so it'll give you some progress messages here, and you can see what it returns is a list that includes the sample and SNP identifiers that it used, the method, and then the GRM object, which is a matrix a square matrix with dimensions of the number of samples in your data. And you can use this matrix as the random effect in your association testing as well. But the method that we actually recommend to instead is to use the PC relate method to get both principal components and the recent kinship matrix separately and include those as separate inputs to your null model. So we're going to go through the process of finding those now. So the first step that we're going to take before we actually compute the kinship matrix is to select a set of SNPs that are in linkage disequilibrium, meaning that they're they're independent SNPs. And that they don't segregate with each other. So the process to do this is, again, using the snip relate package. We're going to open our GDS file. We're going to get a subset of 10 of 100 samples just so that the things will run faster. Uh, and we're going to call the snip relate package, and then we're going to call this snip GDS LD pruning function. We're going to tell it which samples we want to use. We're going to use the correlation method. We're going to tell it what sliding window we want to use. So 10 million is a reasonable estimate. And then we're going to use an LD threshold where of R squared is zero point 0.1. Here, the LD threshold is in units of R, not R squared. So we give it the square root of zero point 0.1. And then it will give some progress messages about what it's doing. It does each chromosome individually. And then it returns the IDs for all of the variants that it selected on each chromosome. So then we're just going to collapse those into a single vector of variant IDs, and we're going to call it pruned. And these are the variants that we're going to use for the rest of this exercise. Uh, So now that we have our list of variants that we've selected, we're going to use the King robust algorithm to identify kinship between samples. Um, so the method that we're going to use is called King robust. King is a separate piece of software that you can use independently, but it takes Plink files as input. So it's a little more convenient to use the implementation of the King algorithm in the Genesis, in the SNP package, which is called SNP GDS IBD King. Um, so this algorithm is robust to discrete population structure. It's not the best method for ancestry admixture. If you have a sample set that has a lot of ancestry admixture, the standalone King software has other algorithms that are preferred in that case. But we're actually going to take advantage of the fact of a particular feature of the King robust algorithm, which is that it gives negative um, kinship estimates for samples that are very ancestrally different from each other. And that's actually how Genesis is going to select the optimal set of unrelated samples. So we're going to call this algorithm, giving it our list of sample IDs and then our list of prune SNP IDs. And then this will return a list where IBS0 is the proportion of variance for which the pair of individuals shares zero ideals identical by state. And then kinship is the kinship estimate between each pair of individuals. So these are matrices here. So we can extract the matrix element from the King results. We can set the column and row names to be the sample ID. We can look at the dimensions. We have only used 100 samples here, and these are the first few values from our matrix. We'll note that the diagonal values are 0.5. If we wanted to use these estimates in an association test, we'd have to multiply them by 2 so that we'd get ones on the diagonal. And we'll do that later. So we're going to use this function called snipgds IBD selection. And all that does is it takes the matrices of kinship and IBS0 and decomposes them into pairwise kinship estimates. So instead of matrices, we have a data frame with the two identifiers for each sample and then their values. And then we can plot these, and then we get this. So here we can see some relatedness in our sample. So this little cluster here is parent-offspring pairs. So these are samples where their kinship estimate is 0.25 and their identity by state estimate is 0. So there's no alleles that are not identical by state for parent-offspring. Um, and then these are full siblings right here. And then these are second degree relatives down here. And we can see the negative kinship estimates that Genesis is going to use to select unrelated samples. So the next step that we're going to do is PCR in order to get principal components. So we're going to select a set of samples that's ancestrally divergent from each other. So we're going to use this function called PCR partition. And I'll note that um, there's a single function PCR that will do the several steps that we're about to do all together. So it's more efficient to run it that way. But I'm going to step through the individual functions here just so you understand exactly what the PCR function is doing when you use it later. So the PCR partition function takes the king matrix that we just constructed above from our king kinship estimates, both as the kinship object, and we're going to use the same matrix both for kinship and divergence here. And then we're going to set the thresholds for above which a sample is considered to be related. So here, and the kinship threshold that we're selecting is going to define anyone less than first cousins as unrelated. Uh, and we can set our divergence threshold to be the negative of that. And so PCR will partition the samples into unrelated and related sets and just return two vectors of sample IDs, one called rels and one called unrels. And now that we have our unrelated set, we can run PCA, and we're actually going to use a snip relate function, snip GDS PCA, to do that. And we're going to tell it that the sample IDs we're using is the unrelated set. And again, we're using the pruned snip IDs. And it will return this PCA result. And now, so we have PCs for all the unrelated samples, but we also want PCs for the related samples. So we're going to project those values for relatives. The first step to doing that is this function called snipgds PCA snip loading. And for that, all you do is you pass it in the unrelated PCA object, as well as the GDS object, and it gives you the snip loadings for your PCA result. And then to get the PCs for the related samples, you call PCA samp loading, passing it the snip loading, the GDS object, and then the sample IDs that you want to project. And then you can combine the eigenvectors from both the unrelated set and the related set into a single data frame. So now that we have our principal components, we want to know which principal components are ancestry informative, because that'll help us decide how many principal components we're going to include as both for the PC relate algorithm and also as covariates in our association tests later on. So we're going to load an annotated data frame that has the populations of our 1,000 genome samples. We'll check the metadata for our annotation, and we can see that this population here contains the abbreviation for the population. And so now we're going to make a parallel coordinates plot, and this plot is demonstrating the values of the principal components uh, all together on the same plot for a large number of PCs. So you can see how the PCs separate different populations. And here's the code for creating a principal component, a parallel coordinates plot that you can reproduce later. So you can see that the first two principal components separate these populations by color. And then after the first two PCs, we can see that only a couple of samples in each PC are pulled out. If you are using much more samples than 100, you'll get many more samples that there are many more PCs that are going to be ancestry informative, but since we have such sparse data, we're only getting the first two in this case. So we're going to take those first two principal components and we're going to use them to compute kinship estimates adjusting for ancestry. So for the PC relate function, we need to create a seqvar iterator object. So we're going to reset our filter from the GDS object that we were using previously. We're going to create a seq data object. We're going to set a filter for our object to only the pruned variants. And so from now on, everything that we do using our seq data object is going to select only the pruned variants, and it will tell you how many variants were selected here. Then we're going to construct an iterator, like we did last week. And now we're going to call the PC relate function. And we're going to tell it that the training set it's using is the set of unrelated samples. But we're going to include all the samples because we want to get the entire set of samples that are included into our final result to get for kinship. Uh, and then the output of the PC relate function is two data frames called kin between and kin self. And so we can use, once we have our PC relate object, we can use this function called PC relate to matrix that will give us a kinship matrix out of these data frames. So we'll call that function and get a PC relate matrix. And then we can use this for another round of the PC error loop. And so in this case, we're going to, instead of calling each of those functions that we just saw individually, we're going to call the PC error function. And we're going to give it our seek data object where we've reset our filter. We're going to use the PC relate matrix that we just constructed as our kinship object. But we're still going to use the original king matrix as our divergence object because this has those negative estimates that Genesis uses to calculate ancestry divergence. We're going to tell it which samples we want to include. We're going to include just our pruned SNPs again. And then it returns a PCA object. From this, the key thing that you'll want to get out of here is the vectors. So this is the eigenvectors for your PCs. And so here we're just joining our PCs with our annotations, so we have them all in the same data frame for plotting. And now we're going to plot the PCs that we got, and we can see that the PCs are separating our samples by population because these dots here are color-coded by population. And now you can take those revised PCs and use them to compute new kinship estimates. And so we'll just call PC relate again, using the new training set of unrelateds that we just computed. And now we're going to plot the kinship estimates from PC relate. And we'll notice that here we don't have all those very negative kinship estimates that we were getting from the king robust algorithm. All right, in the interest of time, I'm going to skip this pedigree comparison section, but you can look at that later. There's also some advanced notes if you have very large sample sizes. There are some modifications to this we would suggest because this algorithm can get very computationally intensive for very large sample sizes. So if you're working with tens of thousands of samples, this section has some suggestions for how you can do this in a more computationally efficient manner. All right, and it looks like there's I'm going to take a quick break to look to see if there's any questions before we proceed to the next step. So I should click on the frame pad. No questions yet. All right, so we'll proceed with the next step, which is mixed models. And so now that we've computed our kinship matrix and our PCs, now we're actually going to include those in our null model so we can use them as covariates in our association test. So as we did last week, we're going to load our sample annotation. This is our sample annotation file. We're going to need the biobase package for the annotated data frame object. And this is our annotation object. And then we're going to load our principal components. And we're going to select the first 5 principal components to be covariates in our association test. You would generally do this by looking at the a parallel coordinates plot like we just saw. This is with the full sample, not just for if you do the full sample and not just the first 100 people, then five principal components is going to give you a better separation in populations than just using the first two principal components. We're loading the dplyr package so we can join the annotation object with our PCs. We'll save that. Then we're going to load our kinship file, and we're going to use the PC relate to matrix function again. And here we're going to use the scale kinship equals 2. Um, so we need to multiply our kinship estimates by 2 in order to include them in our null model. Because we want our values on our diagonal here to be around 1. And so we can see that after we've scaled everything by 2, and we look at the beginning of our kinship matrix, these values on the diagonal are close to 1. And so now when we fit the null model, we're going to select only samples that have that are in our kinship matrix. We're going to set our outcome. Our covariates are going to include the first 5 principal components here. And then we have this new argument called covmat for covariance matrix, and we're going to give it the kinship matrix for the covariates for the covariance matrix. Like we did before, we're going to set our grouping variable to be the study, and then we'll compute the null model, and we'll save it for later. And now we're going to run an association test using that null model. So just like last time, we're going to open our GDS file, create a seq data object, create an iterator out of our seq data object, and then we can just run assoc test single using our iterator and the null model we just computed. And it will give us some association test results, and we can do a QQ plot of those. And here we see that the all of our points are around this red y equals x line. So if you remember from last week, the points were kind of above this line, and that indicated that there was something going wrong with our association test. And the thing that was going wrong was that we were not correctly accounting for our population structure and kinship in our analysis. So now that we've included kinship and population structure in our analysis, our association test is much better behaved. So this is what you want to look for when you're doing an association test, is that most of the points, your p-values should lie along this line of observed versus expected p-values, and you only want to see a few points up here. We don't have any real signals in this particular association test because we just made up our outcome variable. But if you have a real signal, it should diverge fairly far down the graph and not way down here. All right. So the next section that we're going to look at is for how to include variant annotations in our tests. So we're going to start by using some bioconductor annotation resources. Um, and we're going to need our null model that we've already computed just now. We're going to need our sample annotation, and we're going to need our GDS file. And then we're going to load some packages that Bioconductor uses for variant annotation. So there's the genome info DB package that we'll need. And we also need the genomic ranges package and this TXDB human genes package. So we're going to get the ranges from or the variants from our GDS as a GRanges object. And so if you remember from last week, this GRanges object represents all of the variants in our GDS file as ranges. So it's either a single position if it's a single nucleotide variant, or if you have an indel or other type of structural variant, you could have a range of positions for your variant. And now we're going to load this transcript database package and just take a look at what this object is. So this is a data from the UCSC genome browser. It's in build HG19 to match the example data we're using. So whenever you're working with annotation, you want to make sure that your genome build matches the data that you actually have genotypes for. It's a human organism. There's various other information about the data in this package. And so you can take a look at the what the transcripts look like. So it gives you the chromosome, the position range for each transcript, and then the name of that transcript. And so we'll notice that this chromosome names here have a CHR prefix before in front of them, whereas ours in the GDS file did not. So in order to match up our GDS variants with this transcript object, we need to rename our GRanges object so that it has this chromosome prefix at the beginning. So we're going to use this rename seq levels function for that. And so now our GRanges object has matching names for chromosomes. And once we do that, we can use this function called transcripts by overlaps, which extracts from this database of transcripts all of the transcripts that overlap with a variant in our GDS file. And we also want to get the gene ID column out of the transcripts. So now, after we've done that, these are all the transcripts that overlap with our GDS file, including a gene ID. So just as a simple example, I'm going to define genes as overlapping transcripts here. This isn't probably not how you would do it in reality, but for the purposes of this exercise, we're just going to take this transcript and we're going to call the reduce function. And what that does is it takes all of the transcripts that overlap with each other and make them into a single transcript with the earliest position and the latest position as the boundaries of that transcript. And now we're going to rename our sequence levels back to match our GDS file again, since we need to use these to find variants in our GDS file. And so we're going to end up with this GRanges object that I'm calling genes here. And each element of the GRanges object is going to be a range. And we're going to use these ranges as input to our burden test. And so, as if you can remember from last time, a burden test is selecting a number of variants in a region and testing them all together for association under the assumption that maybe each variant in this region might have a small effect, and if you aggregate them together, you can detect an association signal for rare variants in the genome. So we're going to define an iterator object, and here, last time we just ran sliding window tests, where we just ran a sliding window across the genome to try and estimate our burden test. But here we're going to define specific ranges that we've just constructed above. So we're going to construct a range iterator. And then we're going to call it on our seqvar data object, and our variant ranges is going to be this genes GRanges objects that we've just defined. And then we're going to set our maximum allele frequency to be 0.05. In reality, if you're not doing this on test data, you'd probably set this lower and run a burden test using the associate aggregate function. And this is what your results look like. It gives you a p-value for each variant. The variant info field here will give you the variants that were included in each range. You might occasionally have ranges that have no variants in them because you did that filtering on minor allele frequency. So you might have even though we base this on overlapping ranges, we might be filtering out some of them with our allele frequency designation. And So the next thing we might want to do is use a different type of annotation in order to select variants. So I just showed you an example using bioconductor annotation, but there's a lot of other annotation software that you can use, such as ANNOVAR or WGSA. If you look at the slides on variant annotation that are linked from the class notes, there's some information about various annotation packages that you might want to use in order to annotate your genotypes. So we're going to assume that we've run one of those pieces of software and that we have some parsed annotations in TSV files, and we've split them into SNVs and indels. And we're going to load both of those and combine them together into a single annotation object. And we might have something that looks like this. So we have chromosome position, ref and alt alleles, an ensemble gene ID, a consequence, which tells us what type of variant we have, and then the CAD Fred score, which gives an estimate of like what the impact of this variant is expected to be. So you might want to filter your variants by only ones that are associated with a gene, and then you can group them by gene. You might want to filter on this CAD score. You might say filter variants to keep only ones with a CAD score greater than 3 that are predicted to be particularly consequential variants. Um, you might want to keep intronic variants, and then you can look at what that would look like, and you would get 592 rows for that particular set of filters. So in order to actually use this annotation in Genesis, we need a data frame that has variables called group ID, chromosome position, reference, and alt. And so we're going to transform the data frame we had above into the data frame that is going to be used for Genesis. So we take our combined annotation, we only select variants that are in a gene. We group them then by the gene ID and set our group ID to be this VEP ensemble gene ID. And we can look at how many variants are included in each aggregation unit. So we can group by the group ID or the gene ID, and then summarize how many variants are in each group, and just make a histogram of how many variants you're including in each aggregation unit. And now we can proceed with running an association test on this variant annotation. Uh, So we're going to take an example file, and for this exercise, we're going to include variants from all the chromosomes, um, and we've also defined each unit to include SNPs with in flanking regions around each gene. Our positions are in genome build HG19 to match with the 1,000 genomes data that we're using. And we haven't done any filtering by CAD score or consequence here. In each row of the data frame is going to have the information about the variant and the group ID. And we'll also note that it's possible to assign the same variant to multiple groups. So a variant could be associated with multiple genes. Um, and we'll take a look at that here. So this is what uh, we're going to load this file called variants by gene. And here we have a group ID, that's the gene name, the chromosome, position, reference, and alt for each gene. We can look for variants that are present in multiple groups. So the aggregation unit grouping by chromosome and position, and filtering only variants that are in more than one group. And you can take a look at this here. So here's an example of a variant that's present in multiple groups. So it's the same chromosome position, ref and alt, but it has two different group IDs here. And so for this exercise, since we're defining each group separately, instead of just a simple GRanges object, we actually need a GRanges list object because we can have multiple ranges in each unit. So we're going to define what's called a seqvar list iterator object. And in order to construct this object, there's this function called aggregate GRanges list, which is going to take as input a variant data frame that has this format, and it's going to return the GRanges list object that we need as input to our uh, seqvar list iterator constructor. So it takes the variants. It finds all of the unique group IDs, and then for each group ID, it selects only the variants in that group, and then it creates a GRanges object with the variants in that group. And so what we get out of this is a list of GRanges object. So now that we've constructed this, we can call the aggregate GRanges list object function on the variant data frame that we just constructed. We find out that it has 127 GRanges objects in this list. And these are their names. This is the first one. It's got a single range in it. And so now we construct the seqvar list iterator on our seq data object using this aggvar list as our variant ranges. We need to load our null model that we constructed earlier, and now we're going to run a burden test on this object. So we need our iterator object, we need our null model. We're going to tell it that we're running a burden test, the maximum allele frequency we're considering, and then the weights that we're assigning to each variant. And then we can look at the results where we do have some missing values for some of the ranges, but some of these are returning p-values. And we can construct a QQ plot that has the result of our burden test here. And there's an exercise that you can do that is to use um, a different GRanges list object where you can construct the units based on the position rather than the name of the gene. Since we're working with a subset of data, most of the genes in the group ID have a very small number of variants. So this is another function that you can use to construct an aggregate GRanges object using a seqvar range iterator, which is just based on position. And so you can try this after the class to run a scat test using this object. All right, and so that's the end of my prepared material. So I'm going to take the rest of this time to answer questions. I saw one question in the chat about the criteria to select PCA. So I'm guessing that means how many to select how many principal components you would actually use in your association test. Um, for that, you would use the best way that I have found to do that is to use the parallel coordinates plot. So if we go back to that section of the notes and scroll down, this is the parallel coordinates plot where it shows the distribution of values for each principal component across some number of PCs. And if you make this plot and color code by known populations of your samples, you can see where in the plot the populations are separated um, uh, by the PCs and where it looks like just individual samples or noise is happening. So in this case, it's only the first two PCs that are being pulled out. Let me see if I can find a good example of like a real one of these plots. Um, let's see. Um, this is a paper on the Hispanic Community Health Studies study of Latinos. And I believe we published a parallel coordinates plot. Yes, here we go. So this is using, oops, let's zoom in a bit. So this is using real data. And so this shows you what a real parallel coordinates plot might look like if you're using real data. This was from a study of Hispanic Latino individuals in the United States. And they self-identified as one of six different groups. And you can see the group names here. And so we've color coded by the group names. And you can see that in this case, the first five principal components are clearly separating groups of different colors. And then after the first five principal components, it just looks like noise in the principal components. So for this study, we selected the first five principal components as the number that we were going to use when as covariates in our association tests. All right, let's see if we have some other questions in the chat. Um, Correcting for population structure. So what are additional considerations where genetic diversity is quite high? Um, I think that's a good question. I mean, I haven't personally worked on African data sets on their own before. So, and I think the same principles would apply, but I expect that you might see much more complex population structure for working in African data sets. But you can apply the same principle of looking at the parallel coordinates plot and seeing if you know something about where your different samples are coming from, seeing how many PCs are separating known population groups in your studies. Um, how much PVE do the selected principal components explain? Um, that is a good question. I think it really depends on your data set. I mean, it can be, you can look at, you can calculate the percentage variance explained using some of the elements that get returned from the PC error function. Let's go back and look at the PC error output. I think that was actually down. Okay, yeah, here. So the if you get the PC, if you get your output from PCR, you can look at the variance explained with this var prop element from your from the PCR output. What is the advantage of the Genesis package over GAPIT or RMVP? I'm not familiar with those other packages. I think that um, Genesis is not the only package out there, so you can probably get good results with other packages as well. The thing that I think Genesis does that's unique is it separates out the recent kinship from more distant population structure, so you can include those effects separately in your association testing. Um, If we do LD pruning before GWAS, what about the loss of important markers? Oh, so for the LD pruning is only for the kinship and population steps. So when you go back to do your association test, you would include all variants in your association test, not just the LD pruned variants. So you would use the prune variants to get your principal components and your kinship matrix, but then you would reset the filter on your seq data object and include all the variants in your association testing. How do we interpret the kinship plot and the QQ plot? Um, actually, let's maybe we have since we have a few extra minutes, we can go back to this section on comparing with a pedigree. So this is one useful thing about getting the kinship um, matrix is that if you know something about the relatedness of your samples, you can use this as a quality control measure to compare what you expect the relatedness in your samples to be to what's actually observed. So there's a function in the GWAS tools package that's called pedigree pairwise relatedness, where if you have a pedigree file, you can get expected pairwise relationships based on that pedigree. So this is what a pedigree file looks like for it has a family ID, an individual ID, father, mother, and sex. If you call this function on that pedigree object, it'll give you a table of relative pairs, and you can look at what you expect the relationships to be between everyone in your study. Uh, and so here are all of the designations. PO is parent offspring, full siblings, half siblings, grandparent grandchild, avuncular, half avuncular, and then everyone else is considered unrelated. Um, and so here's an example of assigning um, relationships to expected relative pairs, or to. So you can take your table of expected relative pairs, and we're going to characterize these as degree 2 and degree 3 if they fall within different ranges of kinship. And now we're going to assign degrees of relationship to observed relative pairs. And so these are different thresholds you can use for assigning different degrees of relatedness. And so you'll notice that it's a series of powers of 2, where duplicates are going to be anything that's greater than this level of kinship. And then 3, 5, 7, 9 goes to degree 1, degree 2, and degree 3. Um, and so here we're just setting each of for all of our observed relationships, we're using the kinship values that we computed from PC Relate and assigning them labels based on what their degree of relatedness is. We also need to have a K0 cutoff to distinguish parent offspring from other first degree relatives. So once we've done that, we can get a table of observed relatives, and then you can plot those and get something that looks like this, where we've color-coded the observed relationships of all of our kinship pairs. So this group up here is parent offspring. This group here is full siblings. Um, this group here is second degree relatives, and this group down here is third degree relatives. So when you're looking at a kinship plot, you can look at the different regions of the plot and see where people are lying in the plot and determine their relationships based on those plots. So if you go up here, once you get a sense of where all of the different relationships are in the plot, if you look at a plot like this, you can say, okay, these people must be parent offspring, these people must be full siblings, etc. And then I think there was a question about interpreting QQ plots. So when we looked at our initial single variant tests without including population structure and kinship, So we can see how this is the y equals x line for when the observed and expected p-values are the same. And we can see how pretty early on in this plot, the points are above the observed line. So that means that our p-values are higher than we would expect them to be. And this is indicating that something in your association trust isn't being modeled properly. But when we include the kinship and population structure in our association test, as we did here, where everything else is the same and the only thing that we've changed is including the PCs as a covariate and the kinship matrix as a random effect in our null model. Now we can see that the points, the observed p-values are right on this line. And so this QQ plot looks like it's doing a much better job of modeling our association. Can Genesis be used for haplotype analysis? Is it required to use phase data only? So Genesis doesn't take into account the phase of your data. So your data can be phased or unphased. It won't make a difference to Genesis. It just considers the difference, the two alleles independently, and it doesn't account for the phasing between them. So you would have to use different software to you include phasing in your analysis. Uh, Explain the CAD score for U greater than 3. I'm not an expert on variant annotation, but my understanding is that CAD scores are an estimate based on the annotation. So is it expected to be a deleterious variant or is it expected to be a benign variant? And so you can choose a CAD score threshold based on how impactful you want the variant to, how impactful the variant is, and what threshold you want to set for that. I think probably would depend on your analysis. 3 might just be an example of a value that some people might use in an association test. Um, what is the solution to reduce the QQ plot inflation? I think I would, if you have a QQ plot that's inflated, I would review what covariates you're including in your analysis and see if there's any confounding effects that you might not be accounting for. Like, did you include like age and sex as covariates? Did you include population structure or PCs as covariates? Do you have relatedness in your samples that maybe you weren't aware of, and so you need to include a kinship matrix in your analysis? Um, and I'm not sure I understand this question. Does kinship and QQ plot decide the downstream analysis of the left GWAS part? So sorry, I'm not sure what to say about that one. All right, it's at the top of the hour, and so thank you all for joining me for this session. And the website is going to stay up indefinitely, so feel free to go back on the website and look through all of the materials and try out the exercises on your own.
Thank you, Stephanie, for two wonderful workshop sessions. We appreciate your time and commitment to um, building capacity in GWAS analysis for our community, and we are very thankful. So everyone, we will have our next webinar session next month on August 12th. It will be on multi-omics data integration, and we'd love to have you join as well. Stephanie, can people reach you directly if they have questions, further questions on GWAS?
Yes, here I'll put my email address in the chat. So you can email me if you have questions on using Genesis, and I will do my best to answer.
All right. I will also post that on the Framo pad in case you'd like to jot it down somewhere. Again, thanks everyone. I know it's quite late here in Kenya, and we are still thankful that everyone joined. Please, when we share our feedback survey, please answer it so that we can make the sessions even much better for you and for your research needs and for the questions that you'd like answered. Also, be on the lookout for the next webinar that we'll be sharing. Thank you so much. Bye.
Bye bye.